Hi, today we will be discussing about origin of species. It is estimated that world over we have a species between 7 million and 15 million. So many species are there, and each and every species is having a unique function to play, because it is having an interaction to play, and because of that thing other species also existing. So it's a very important thing. Species are very important thing as far as the biodiversity is concerned. And this particular lecture on origin of species is important from the perspective of speciation, origin of species, and also the extinction. So the world over, if you see, we have seen a lot of extinctions, and we are in the midst of 6th extinction. The past extinction has taken place in the Ordovician period, Devonian period, Permian, Triassic and Cretaceous period. And during that period the extinction has taken place, but the extinction that has taken place in the past and the present extinction is different. The present extinction is the 6th extinction. What is the difference? The difference is that the past extinction was driven by the natural causes, but the present extinction is driven by the human beings. And second difference is the extinction rate. The extinction rate in the past was less, but now in the present extinction the rate of extinction is 100 to 1000 times more than the background extinction. So look at it, how important it is, how important the extinction is taking place, and in that sense, in that perspective, this origin of species or speciation is important. What exactly species are? Species, quote unquote, it means a group of organisms that because of close genetic and physical similarities can naturally mate with each other and produce viable offspring. The important aspect in species definition is that intermating, interbreeding. The species is a group of organisms that has to interbreed. That is a key important thing. And this species is very important. Why? Because it shows, it exhibits a level of integration. And this understanding about species, speciation is important for further understanding in pure biology, for understanding the diversity of organisms, for understanding the species diversity, genetic diversity, ecosystem diversity. And that is the reason why this species concept is very important thing. And very important that because we are in the midst of extinction, so we need to conserve the species, and for that to take place we need to understand what exactly the species are, what is the concept behind it, and how it is evolving. So that's a very important thing. So there are many concepts as far as the species are concerned. There are many concepts, but the foremost concept, biological concept of species, is very important thing, because there are other concepts also. Reproductive species concept, recognition species concept, it is existing, but this biological concept of species is very important thing. Way back in 1809, J.B. Lamarck came with the wonderful definition on species. He was talking about the environmental condition, he was talking about a biological bond that is existing between the individuals of the same species. The biological bond, it means that each and every individual will be recognizing the other, and after recognition the individual will be intermating. So that is a key thing. These are the three important things he stressed, J.B. Lamarck, way back in 1809. Later many people have given a conceptual, even the Darwin also was talking about it through his origin of species. He has talked in detail about origin of species in terms of adaptation. But Ernst Mayr in the year 1969, he came with a wonderful concept, that is biological species concept. It is a very important concept. Based on that thing we can understand further the process of speciation, what it means. What he said that these species is a group of interbreeding natural populations that are reproductively isolated from other such groups. It's a very important thing. He was talking about reproductive isolation, and he was talking about that the species consists of individuals which share a common gene pool. That's a very important concept that is said through the common gene pool concept he has given it. And I told you that these species, because they have been isolated, reproductively they are isolated, and the species do not interchange genes, gene flow will not be taking place. That's a second important concept, underlying concept in biological species concept, if you mean it. So this particular thing is very important thing, because this biological species concept has helped to identify or recognize a species. Otherwise it will be difficult. I will take an example of Drosophila, pseudo obscura and Drosophila persimilis. Both Drosophila were morphologically similar. It's very difficult to indistinguish them. It's very difficult. So in that case also what happened? They had a concept this biological species concept that helped even this species which was morphologically similar, and they have divided this species Drosophila pseudo obscura and pseudo persimilis. How? By the concept of interbreeding, because the species, two species will not interbreed, individuals within the species will interbreed, and based on that concept they have classified these two, Drosophila pseudo obscura and Drosophila persimilis, into two different species. It is a very important thing. And what are the criteria? I have talked about species. What are the criteria for recognizing the species? There are three important criteria. One is the phenotypic criteria, second is genetic criteria, third is reproductive isolation. We will go one by one. First is phenotypic criteria. It deals with the morphological features. What the difference existing between the individuals or between the species, what are the features, that is morphological features. So based on the morphological features we will be categorizing the species. That is a very important thing, because at the laboratory condition we can understand that if there is a morphologically they are different, that means that we can categorize the individuals in two different species. That is a very, very important thing. This is the foremost concept, foremost criteria. Along with that thing, biochemical features, physiological and ecological features also have been incorporated. By using these all the features we can recognize the species. But of course there are certain drawbacks. What are the drawbacks? For example, in the male and female individuals of sexually dimorphic species would be classified in different species. It is a very, very difficult thing, because in the case of sexually dimorphic species, that is different forms, that is male and female have different forms. So they will be trending to classifying those two individuals, male and female, in two different species. It is wrong. So that is one important drawback as far as the morphological features are concerned. Another example is in case of insects, even the developmental stages in the life history, if you see, there are different developmental stages. Those developmental stages also will be unfortunately may be classified as different species if we go for what? Morphological features, if we go for phenotypic criteria. But still this morphological criteria or the phenotypic criteria is having lot of positives. The first and foremost positive is, this is the important criteria, we are using it to understand the fossils, to categorize the fossils. We are using this phenotypic criteria. Second important thing, in case of asexual organisms, we have to go for phenotypic criteria. Why? Because I was talking about the reproductive barrier, I was talking about the reproductive isolation. So in this case, asexual organisms, there is no isolation, reproductively it is difficult because it is asexual in nature. So in that case we have to go for morphological features only. So these two are very, very important examples wherein phenotypic criteria has been used for recognizing, for categorizing the species. Next is the genetic criteria. Genetic criteria is a very, very important criteria. To start with, I was talking about the reproductive isolation. So let me explain this thing. Individuals of the same species should be genetically more similar. Individuals of the same species should be similar in nature. The genetic similarity has to be understood, and we need to understand that thing by using a measure, and that is a basic thing. Only when we have a robust measure for understanding the genetic similarity, then we can go for recognition. There are many methods available to measure the genetic identity and genetic distance. Genetic identity is a very simple concept, and for example, the genetic identity, if it is unity, if it is 1, that means that the two populations have the same alleles at identical frequencies. If it is 0, in that case the populations, two populations will not have the same alleles, it is different. So this genetic identity is a measure, and it is used in case of this particular criteria, genotypic criteria. So by using this genetic identity we can measure the genetic distance. It is a very simple thing, it is a negative logarithm. If it is a natural logarithm, it is just simply negative natural logarithm of genetic identity. If it is a common logarithm, it is minus 2.3 common logarithm of genetic identity. It is a simple concept. What it means? Genetic distance is a measure of average number of allele substitutions accumulated per locus. It is a very important criteria. By knowing this thing, by simple calculations of the allele frequency, we can calculate the genetic identity and genetic distance. By this way we can categorize, we can recognize the species correctly. The third important thing is the reproductive isolation. So when I say this reproductive isolation, this is the foremost important criterion. And based on that thing, there are a lot of mechanisms. For reproductive isolation, there are a lot of mechanisms that is involved or that is aiding in reproductive isolation. The mechanisms can be pre-zygotic or post-zygotic. When I say pre-zygotic, it is before the mating, and when I say post-zygotic, it is after mating. So when I say there are a lot of criteria, let me see one by one. The pre-zygotic mechanism, the first and foremost mechanism is habitat isolation. When I say habitat isolation, that is the species occupy different habitats in the same geographical area. The species occupying different habitats in the same geographical area. So by that way, what happened, since it is occupying different habitat, we can categorize the species, we can recognize the species. Let me give an example. The example is the mosquito, common Anopheles mosquito. There are a lot of species. How? By using the habitat we can categorize. For example, Anopheles maculopennis, it is the larva of this Anopheles maculopennis thrives in a running fresh water. Anopheles melanum, it is found in stagnant fresh water. Anopheles labranchi, it is present in the brackish water. See how habitat is changing in the same geographical area. This is the best example for habitat isolation. And in addition to this thing, these mosquitoes, or for that the organisms, may be differing in prey preference, food preference, habitat preference. So this is a very important criteria. Based on that thing, we can say that reproductive isolation, how important is to categorize the species. The second important thing is the seasonal isolation. Seasonal isolation is a very important thing which encompasses many features like time of maturity, time of flowering, mating, and so on and so forth. Based on these features, we can find out whether the species is different or not. Let me give an example. That in case of toad, the scientific name is Buffa americanus and Buffa fowleri. Buffa americanus, the toad, breeds early in spring, but Buffa fowleri breeds later in the spring. So this is an important difference, isolation, seasonal isolation. The second example as far as the plant is concerned, Pinus radiata and Pinus muricata. In case of Pinus radiata, pollen shedding is taking place in the early February, but in case of Pinus muricata, the pollen shedding takes place in the month of April. So by that way, their kind of isolation is taking place. That isolation is because of season, temporal isolation. Next important thing is the sexual isolation. Sexual isolation means it refers to a mismatch in the behavior or the physiology of the organisms, and it also includes what? Species specific courtship. The best example is in case of butterfly and moth. There are a lot of examples, but the best example is butterfly and moth, which exhibits a very important character, that is sex attractants. By sex attractants, it will be finding out the particular species. So that way, what I wanted to say is that sexual isolation is also a very important part of reproductive isolation. The next one is the mechanical isolation. When I talk about mechanical isolation, it means that the mechanical structure of reproductive organs that prevents cross fertilization between species. I'll give an example, wonderful example, that is orchid. Orchid is it should be pollinated by a specific bees. It's a very interesting example, because in case of bees, if you see these orchids, what happened, the female flowers will try to attract the active bees. And how it attracts? It attracts with its own color, that is female flower color. So it looks like as if it is like a female bee. So because of this color and the structure, the reproductive organ structure, these bees, active male bees, will be attracted, and in the process it will be pollinating. See how important it is. Mechanical isolation, the best example in case of orchid and bees. Next is the gametic isolation. See, the gametic isolation means we can very explicitly mention the examples of marine organisms and flowering plants. When I say marine organisms, the attraction between the sperm and the egg, that is reduced if other species are involved. That is as far as the marine organisms are concerned. But as far as the flowering plants are concerned, the gametes, both the male and female gametes, their interactions, that also will be different. So there is a lot of incompatibility that will be existing between the male and female gametes as far as different species are concerned. So these two are the very important examples as far as the gametic isolations are concerned. I was talking about the pre-zygotic mechanisms, but we will go for now post-zygotic, that is after mating. The first and foremost thing is hybrid inviability. So the hybrid formed after the mating, that is after the hybridization, the hybrid that will not be exhibiting any viable nature. So why? Because the zygotic development itself fails. Because of that thing, hybrid inviability is taking place. There are a lot of examples. Think about a cross between a goat and sheep. The offspring will be, that definitely will be hybrid, but it will be inviable. Next is hybrid sterility. Hybrids fail to produce functioning or functional gametes. There are many examples. In case of interspecific crosses, in case of plants, I mean to say, there are a lot of examples are there wherein the product of interspecific cross, the hybrid is sterile in nature. It's very difficult, it's sterile in nature. And as far as the animal population is concerned, the best example is the cross between a horse, the female parent as a parent, and ass as a male parent. It results into a offspring hybrid, that is mole. So this is about the example of hybrid sterility, how important it is. There are a lot of examples of hybrid sterility in interspecific crosses that has taken place in the plants and animals as well. Next one is hybrid breakdown. It's a very important mechanism as far as the post-zygotic mechanism is concerned. The fertility of progenies of hybrids drastically reduces. Why? Because this is because of the formation of inferior genotypes by recombination, which is a product of crossing over, which is a product of segregation that is taking place in the non-allelic segregation, I mean to say. So this, because of the formation of inferior genotypes, this hybrid breakdown is taking place. There are a lot of examples for this particular thing. See, I mentioned very in detail about what exactly the biological species are, but still there exist certain difficulties with this biological species concept. The first and foremost important thing is that the existence of species as an integrated gene pool. This is an important feature of this particular concept, but this is not corroborated with evidence. It has not been found properly. So in that way, what I wanted to say is that this is a difficulty. The second important thing is reproductively isolated unit. This is a reproductive isolation is an important criteria. It is an important feature of this biological species. But this particular feature is not seen in the plant. In case of plants, we'll see how it is. In the case of plants, there are many developments. That is polyploidy, autopolyploidy, allopatric. This has developed or led to the development of a lot of crop plants. So what I want to say is that the reproductive isolated unit breaks down in case of plants. That is a difficulty as far as this particular concept is concerned. The third one is the lack of agreement among the criteria. I was talking about all the criteria, phenotypic criteria, genetic criteria, and reproductive isolation. The kind of agreement that has to exist, that is lacking in this particular thing. So these are the three difficulties that we are encountering in species concept. However, this biological species concept is a very, very important concept. This is given us a way to understand the process of speciation. Speciation, we'll see what it is. The speciation, it means that the processes by which new species are established. It's a very important species because through this speciation process, new species will be formed. I was talking about, the start of the lecture, I was talking about that how important speciation or for origin of species with regard to the mass extinction. It's very, very important thing, because when I talk about speciation with regard to the mass extinction, the important point is if the speciation process is more than the extinction, then no problem, because the number of species is more than the extinct species. So that is a key thing. So we'll understand what are the mechanisms, what are the processes involved in speciation. First process is allopatric speciation. It means what? It means that the formation of new species in population geographically isolated from the parent species. Here I am talking about the geographical isolation, and it is a prime criteria. Geographical isolation is a prime criteria for formation of new species that forms the bedrock of allopatric speciation. So when I was talking about the geographical isolation, that forms the first criteria, first important feature of allopatric speciation. This has to be there, only then new species will be formed. And because of the existence of geographical isolation, the gene flow will be prevented. Only when there is a prevention of gene flow, then only new species will be formed. So that is a criteria. And next one is that how it is made possible. So this is made possible by creation of islands of population. Islands of population will be created through establishment of barriers. The barriers can be sea, mountain, valley, glacial masses, vegetation zones, water bodies, so on and so forth. These forms a barrier. Because of this formation of this barrier, the gene flow is prevented. So let me give an example of a water body. In case of freshwater body, lake is a water body, river is a water body. So because of this water body, what happens? This species whatever developed in this particular water body, that will be endemic in nature. That is a very important criteria, because the lake will be having endemic species. Think about a vegetation zone. The dividing line between one zone with the other zone, it will be very sharp. In that particular dividing zone, since it is sharp, this forms as a barrier. In case of a mountain, in case of a valley, valley also forms a barrier for the movement of the organisms, in the land organisms. So what I want to say is that these barriers creates islands of population, and that leads to geographical isolation, and that is the reason why later stages a species will be formed. Second important step as far as allopatric speciation is concerned, that is genetic reorganization and divergence in isolated population. I was talking about the isolation. Because of geographical isolation, the population that has become isolated, this isolate exhibits founder effects. It exhibits kind of severe inbreeding, selfing. Since it exhibits severe inbreeding, that leads to what? Homozygosity. And this particular thing, the inbreeding and the resultant homozygosity, is a very important thing. Why? This results in genetic reformation. It results in a revolution that is called genetic revolution. And this leads to what? It leads to a shift, ecological shift. It means that this isolates wherein it has been completely isolated, and because of the inbreeding process and that result in homozygosity, what happened? A genetic transformation, a genetic combination, that is a variation that is taking place in the change in the genetic combination, and the genetic combination or recombination that leads or provides the power to the individual to thrive in the adapted conditions, new and environmental conditions. That is called ecological shift. Only then we can expect the phenomenon like random genetic drift, and this random genetic drift, which signifies that there is because of the chance factor, because of the random error, there is a kind of drift is taking place, and this is very quite common in case of smaller population. And in this genetic drift, what it leads to? It leads to a platform for the species to origin. Next is the reproductive isolation. See, I was talking about reproductive isolation in detail, but it means what? I already explained it. In a nutshell, I wanted to give that this reproductive isolation is chance byproduct of genetic reorganization and divergence that is taking place between the population. It is a chance byproduct. I would like to stress how allopatric speciation is important, because this allopatric speciation has provided us a way to think that how species have been originated. And there are many examples. I'll take an example of cichlid species, Haplochromis species in Lake Victoria. Lake Victoria is a very famous lake in Africa, and this has been formed, Lake Victoria has been formed way back in the Pleistocene era. And when I say Pleistocene period, it is 7,50,000 years back this particular Lake Victoria was formed. And if you understand the process of formation of Lake Victoria, you will understand further there is, along with this Lake Victoria, there are other lakes also, Lake Albert, Lake Kivu, George, Edward, so many lakes have been formed. So the species present in the Albert, or all whatever the lakes I talked about, plus the Lake Victoria, if you see it, the species are similar. This similarity is very, very important thing, and in this similarity you will be finding one important species that is very famous, that is cichlid species, that is Haplochromis species. So there is a kind of similarity that is in the species present in the Lake Victoria, Lake Edward, Albert, Kivu, George, everything. So it is a kind of similarity that is existing in this particular species. Why it is? The origination, you have to understand the origin of Lake Victoria along with other lakes, and we will need to understand that in the process of evolution, of the Lake Victoria, what happened? This particular lake was evolved way back in the Pleistocene period, but what happened after that thing? There was a lot of tectonic instability. Because of this tectonic stability, what happened? The huge mass water body that has been fragmented into different lakes. That one is Lake Victoria, the other one is proto lakes. I was talking about the Edward, Albert and everything, these are all proto lakes. So kind of fragmented, the lake body was fragmented. This process of formation and fragmentation was taking place concurrently. When I say so many times it has taken place, that was the reason why so many species have been formed. To explain further, I'll explain to you that this particular, when this formed as a huge body, water body, what happened? There was a lot of species that was thriving. Along with that particular thing, when there was a tectonic instability, because of that thing the sea level rise and fall that was taking place. So there many small lakes were being formed, and in the process what happened, in the small lakes also the species was thriving, and it started adapting itself to the smaller lake, and a new species was forming. And again what happened, again the change, so again a new water body has formed and all the species have been combined together. So this process was continuous, and that is reason why for the past 7,50,000 years, if you see, there are more than 200 species have been formed, evolved, and this is the reason why that has taken place in the Lake Victoria, and that is a very good example of allopatric speciation, or indirectly reproductive isolation, indirectly geographical isolation. That is a key thing for allopatric speciation. Next we will go for sympatric speciation. It is a very important thing. On the other hand, this sympatric speciation is a very the importance, though it's very important thing, but not like your allopatric speciation. In case of sympatric speciation, there is no geographical separation between species population. So the species that is there, it is continuously arranged, it is continuously distributed, and in that case also new species are formed. It's a very important thing as far as the concept is concerned. It's a very important thing. There are a lot of examples in case of plants. The involvement of polyploid, I was talking about polyploidy. See, whether it is a autopolyploid or allopolyploid, many species, plant species have been evolved by this particular process, sympatric speciation. The examples of autopolyploid, there are many. For example, the tetraploid potato, tetraploid coffee, these are tetraploids, autotetraploids. So another example as far as the autopolyploid is concerned, in case of watermelon, that's a triploid, autotriploid. So best example in case of autotriploid, what happens is that the watermelon will not have the seed, that sterility, the triploid condition will be sterile, and that is made possible because of this process, polyploidy process. And as far as the allopolyploidy process is concerned, we are eating the Triticum aestium, that is an hexaploid, This particular wheat crop is a best example of allopolyploidy, best example. So what I wanted to say is that sympatric speciation is also a very important thing, which has signified that in the absence of geographical separation also the species will be originating. And the examples I've mentioned, that in case of polyploidy this has been extensively exhibited. To summarize, the species and the origin of species is a very important thing, because when we understand the process that leads to speciation, we can understand that how important it is, how much time it is taking place for the species diversity to take place. And this point should be taken care of by taking into account the extinction rate, by taking into account the human action. So that's the key thing. So we need to understand that speciation versus extinction. So when you understand this particular thing, we will be thinking about the importance of species, we will be thinking about how the threats to biodiversity, how important it is, the drivers, both direct and indirect drivers, and we will be concentrating on the legislative measures, we will be concentrating on the conservation measures, whether it is an in situ conservation or an ex situ conservation. This is a very important outcome. So this particular lecture gives us a thought forward. How important the species, how important the conservation is, how important the services it is providing. And this is the ultimate speciation, the origin of species, this is the ultimate topic. It's a cornerstone of sustainable management of biodiversity. Thank you.